we help you today?
Yes, right this way.
I see. Okay, you take that side and I'll take this side. And then tell me if you find anything immediately.
Okay. I got it.
Oh wait, I found something. Well, this hair sample contains all the DNA we need to solve the case.
What's DNA again?
What am I going to do with you?
Science.
What is DNA, you ask? Well, DNA stands for deoxyribonucleic acid. Before we get into DNA and its structure, let's talk about an essential element of DNA, genes.
All DNA is composed of genes. A gene is a sequence of nucleotides within a portion of DNA that codes for functional RNA. Here's an example of how a gene is represented. The sum of all these genes make a genome. Okay, now where exactly are genes in DNA found?
Well, DNA is found within the nucleus. It's in the form of chromatin. These chromatins come together to form chromosomes in the process of cell division, or mitosis.
Enough about chromosomes. What about DNA structure?
DNA is in the form of a double helix. This helix is composed of nucleotides. All nucleotides are composed of nitrogen bases, sugar, and phosphate.
Now these nitrogen bases are found in two forms, purines and pyrimidines. Purines refers to the nitrogen base adenine and guanine, whereas pyrimidines include thymine and cytosine and uracil. The sugar found in the nucleotide is a deoxyribose.
Okay, let's focus a little bit more on the bases, adenine, guanine, thymine, and cytosine.
If you examine the picture, why? Well, this is what we call base pairing. Base pairing refers to the pairing of complementary bases. For example, it results in the DNA strand having directionality. So we know that the bases are paired together, but what keeps them together and intact?
Let me answer that for you. You see, each base is held together by hydrogen bonds. These loose bonds allow the DNA to undergo any changes if necessary, but also to keep them united to last. The adenine and thymine pair requires 2 hydrogen bonds, whereas the guanine and cytosine requires 3 hydrogen bonds.
So all that stuff makes sense, but...
But what?
Well, if we have the suspect's DNA with us right here, does that mean he doesn't have any more?
What? Of course not. Do you really think people only have one set of genes? Think, apprentice. Have you ever heard of DNA replication?
Yes. Just now at least.
Whatever. Scientists...
What must be understood is that every strand of DNA is in a double helix form. One strand is the mother strand, and while the other is the new strand. These two types of strands are created through the process of DNA replication is the process of producing 2 identical replicas from one original DNA molecule. This process is crucial for mitosis, meiosis, and binary. So this unique process can be broken up into 5 basic steps. Step 1. The DNA helix unwinds into ladder form. Step 2. DNA helicase breaks apart the hydrogen bonds between the nitrogenous bases. Essentially unzips the 2 strands, creating this, a replication fork. Step 3. Now, DNA polymerase helps attach complementary nucleotides to the free DNA strand called the leading strand. Step 4. DNA ligase, also an enzyme, helps to speed up the process to create sugar phosphate backbone for the new strand. And finally, step 5. DNA polymerase goes back through this strand to check for errors and mutations, and if it finds any such flaw, it fixes them. Therefore, we will never run out of DNA, really. The process of DNA replication is crucial to our body because it is needed for the creation of new cells, and DNA transmits signals that create protein for our body. Cells make up the body and prepare our bodies. So, if we didn't have any replication, then we'd have a limited number of cells and they would never be replaced. So, the human body consists of millions of strands of identical DNA. That hair sample only has a small portion of all the DNA within us.
Well, speaking of hair samples, we need to get ours back to the DNA lab. Let's go! Oh.
Okay, so how are we supposed to find the actual culprit with this?
With the use of DNA fingerprinting, of course.
What's that?
DNA fingerprinting? Well, you can call it DNA profiling too. But basically, it's just a technique used by scientists to distinguish between two individuals of the same species. And DNA processing was invented by that guy over there.
What, that guy? No, I mean the guy next to it. His name is Alec Jeffrey. Well, you see, he invented this process of analyzing DNA in 1985, and he was knighted for the discovery 9 years later, in 1994. There are 3 main steps in analyzing DNA, and I'll let him explain it.
Step 1. Cells collected are broken down by complex instruments to release DNA. Step 2. In step 2, the DNA is cut into fragments using restriction enzymes. These enzymes split the DNA into specific base sequence. The sections of DNA are called restriction fragments. Once the DNA is cut, we move to step 3. In step 3, fragments are separated bases of size using gel electrophoresis. The fragments are then injected into wells and an electric current is applied along the gel. Once radioactive material is added into DNA fragments, it produces this. Last but not least, step 4. In step 4, the pattern of fragment distributions are then examined. Back to you guys.
Well, therefore, the DNA gained by electric gel phoresis is used in the forensic science area to compare a suspect's DNA to the DNA found at the crime scene.
Wait, doesn't that mean that someone else could have the same DNA?
Yeah, like a 1 in a trillion chance, but... Okay then, you get some rest. I think I got this case wrapped up. But don't be late, and come straight here tomorrow morning.
Yes, ma'am.
You've all been gathered here today for a very crucial reason. 2 days ago, Miss Pai...
It's Paya Payu.
Oh, as I was saying, 2 days ago, Miss Paya Payu was murdered, and one of you is responsible for this.
What?
Well, would any of you like to confess? Don't give me that look, banana. Just because you're a fruit doesn't mean anything. Humans share 50% of DNA with bananas, so don't think you're off the hook. And you two, don't act like you're not stuffed with guilt. And you! I like your mess.
Oh, thank you, I guess.
Oh hey, where's the trainee? Is he late again? Okay, whatever. Out of the four of you, one of you is guilty. But since no one will confess, I guess we'll turn to DNA analysis. Scientists? Well. It looks like...
Get down. I don't want any trouble.
Calm down. Just put the banana down and no one will get hurt. I should have known it was you the whole time. Your DNA matched perfectly with the strand of hair. Now, I'm giving you a second chance. Just put the banana down and we'll let you go unharmed.
Well, it's too late now.
Congratulations!
Couldn't have done it without DNA.